Hello, everyone, and welcome to this on-demand webinar. I'm Evgeny Glazov, Regional Marketing Manager for Illumina based in Brisbane, Australia. And I'm excited to introduce our featured speaker today, Dr. Andrew Weeks. Andrew is a pioneer in ecological genetics, and he's been a key player in developing and using eDNA for environmental monitoring. With over 25 years of experience and as a founding director of the EnviroDNA, he's uniquely positioned to share insights into the amazing work being done as part of the Great Australian Wildlife Search Initiative. In this webinar, Andrew will dive into the world of eDNA and explain how this innovative technique is changing the way we understand aquatic ecosystems. He'll also discuss how Illumina sequencing technology is helping us identify the incredible variety of species found in these vital waterways. Please enjoy the following presentation by Dr. Andrew Weeks.
As Sam mentioned, my name's Andrew Weeks. I'm a director at EnviroDNA. A little bit of background about myself and EnviroDNA. I started working with Odonata probably about 15 years ago on threatened species recovery programs. I'm a geneticist by trade and do a lot of work around developing genetic tools and applying genetic strategies to help improve threatened species recovery programs. One of those tools that I was involved in developing as a researcher at the University of Melbourne was really understanding environmental DNA and the technology that sits behind it, which led us to form EnviroDNA in 2017 so that we could extend a lot of that research and the underpinnings that we learnt along that research journey out into the broader community, and which has led to this GAWS project that we'll talk about today. So I'm just going to give you a bit of a high-level presentation. There will be a report, which is also just a high-level overview of our findings from the analyses of samples that you all sent in, and give you a bit of an insight into some of our interpretations. So I'll just go on to, recap, I guess, the process of how this project started. So initially there was a sampling design that was developed by La Trobe researchers, Michael Shackleton and Nick Bond at La Trobe University. They developed a sampling plan that was really trying to cover a lot of the different rivers and streams that form the Murray Darling Basin across the various states that the Murray Darling Basin waterways flow through. The idea was to have spatial scale, which is what eDNA is really good at, detecting species right across a large and broad spatial scale. There was a whole bunch of different metrics that went into that sampling plan, but I won't go into that in detail here. Once that sampling plan was developed, there was a website that was constructed by Odonata where citizen scientists could essentially sign up to sites, select a site, reserve it. Once they'd reserved that site and left their details, then an eDNA kit was sent out to that particular citizen scientist. Within that kit, there was essentially 2 samples that you could take from a particular site, which is what we were requesting everyone to take at a particular site. So there was a syringe, which you used to sample the water, and then there was 2 disc filters, which we used to filter the water through and trap the DNA that is found in the water. There was some preservative, which is used to preserve the DNA, and some gloves, which were, the idea was to keep everything as sterile as possible so that there was no contamination going on. There was also some instructions of how to undertake that sampling that went in the kit, as well as a video that was on the GAWS website that people could view to get an overview of that sampling. Once sampling was undertaken, those samples were then sent back to our labs, EnviroDNA's labs in Brunswick, for DNA extraction and for undertaking the data analysis. We screened each sample after the DNA was extracted using what we call a vertebrate metabarcoding assay. And essentially what that is doing is trying to detect any vertebrate species that DNA was on that filter or in that water that was filtered through that filter. We then undertook a library construction in the laboratory and then sequenced it on one of our good partners, Illumina, an Illumina sequencing machine, which enabled us to then detect the different species DNA that was present within that water sample. If we just take a step back to give a broad overview of what eDNA is. So eDNA is essentially any DNA that a species leaves behind in the environment that ends up, in particular, in water or in the waterways that everyone took water samples from. So that can be skin cells, hair, scales, mucus, and a whole bunch of other different secretions that an animal might leave within the waterway. That DNA, as I said, is then brought back to the lab where we DNA extract, or we extract the DNA and then we analyze it using a variety of different techniques. Now, just to be aware, so it's really a snapshot of what is present at that site, or at least what DNA is present at that site when you take that sample. For a lot of aquatic species and semi-aquatic species, you will generally detect them from only 2 water samples if they're present at that site. For a lot of terrestrial species, though, it's very dependent on A, when that terrestrial species was present in the environment, and B, whether their DNA actually ends up in the waterway. For species that live really close by to a waterway or over a waterway, such as a possum or a bat, then their DNA is much more likely to end up in the water. But for species that are not present exactly at that waterway, then it actually takes other events for their DNA to end up in the water. And that might be some heavy rain, which then washes their water into the waterway. The waterways are often flowing, so that means that the DNA then gets carried down that waterway. So just, I guess, to highlight that terrestrial species detection is really going to be a numbers game of how many times you sample at a site and how often that particular terrestrial species is at a site. Whereas for your aquatic and your semi-aquatic, we're much more likely to detect them. And on the slide here, you just have some examples of some citizen scientists that took the DNA samples, or the eDNA samples, from the waterways. The waterways, as most of you will know, some of them are relatively clear, nice fresh water where you're expected to be able to filter quite a large amount of water through the filter, whereas some of the waterways were quite turbid, which meant that a lot of sediment load, which ends up clogging up the filter, which then means you're not able to filter as much water. We can still detect things from small amounts of water, but obviously the more amount of water that you push through, the better the chance of detecting things in the waterway. Just to give an overall high-level view of what we detected after we put it through that eDNA metabarcoding process. So there was 324 sites surveyed, which resulted in 648 eDNA samples that were brought into our laboratories. There was 144 different taxa that were detected from those samples. We had a total of 5,905 detections. We detected 17 threatened species, either under the federal EPBC Act or through state acts for threatened species. And there were 26 invasive species that we detected. If we dig into that a little bit more, what we found was for the various groups that we were targeting, and this vertebrate assay targets fish, frogs, birds, mammals, and reptiles, essentially. For fish, which we expect to detect their DNA relatively easily in a water sample because they're living in that water all the time, there were 41 different fish species detected. 30 were native fish and 11 were invasive fish, and 6 were EPBC listed threatened species. For amphibians or frogs, there were 20 different species detected, all of which were native to the Murray Darling Basin. We didn't detect any EPBC listed frog species, but we did detect the tusked frog, which is listed in the state of Queensland. There were 43 bird species detected, with 40 that were native and 3 that are introduced species. There were 2 threatened species that we detected from birds. For mammals, there were 32 different mammal species detected, 20 that were native, 12 that were invasive, and 4 that were EPBC listed species, and a couple of other species that were listed in various states. For reptiles, we only detected 8 reptile species, and that's not overly surprising because reptiles shed very little DNA, and therefore that DNA ending up in the waterway it can take a little bit of time for it to get to the waterway for a start. They're not shedding a lot of DNA, so we don't find a lot in the waterway, and that includes turtles, which do live within the waterway. Their DNA is also not overly shed well, like a lot of other species. So species do vary in their detectability. I'll just mention that. So what were the native fish species that we detected? And apologies, I just saw a comment come through. I think it's taking a little bit of time for these slides to click over for everyone to see. So I might be talking to a slide before it actually comes up on your screen, but hopefully we're mostly on the same page. We detected 6 EPBC listed threatened species: silver perch, flathead galaxius, macquarie perch, trout cod, Murray cod, and southern pygmy perch. Some of the more common native fish species that we detected were Australian smelt, which was found at a large number of sites throughout the Murray Darling Basin. I think there was around about 47% of all sites had Australian smelt within it. There's the mountain galaxid species complex we detected. So the mountain galaxid, which is Galaxius olidus for those who know scientific names, that was mostly detected, but there are also a couple of other mountain galaxid species that we can't differentiate from the olidus species that were also detected at a variety of sites. There was 100 sites in total where we detected mountain galaxias, and golden perch were detected at 132 sites, so about 41% of the sites across the Murray Darling Basin. Other native species that we detected grey, so 6 different listed species. Grey-headed flying fox, which is EPBC listed as vulnerable, was detected at 6 sites. Platypus, which is only listed in Victoria under the FFG Act, that was detected at 52 different sites across the basin. We detected the EPBC listed koala, both in New South Wales, Queensland. They were detected at 4 sites, 1 in Queensland and 1 in each of New South Wales and 2 in Victoria. We detected at 1 site the endangered broadtooth rat. We also detected at 1 site a greater glider, which is also endangered. That was detected in the King River in Victoria. And as I mentioned a bit earlier, we detected a single threatened frog species, which is vulnerable in Queensland, called the tusked frog. We detected a number of terrestrial species that are quite commonly found throughout the Murray Darling Basin. One of those was the common brush-tailed possum, and that's largely because they live a lot around waterways, in trees in waterways, and their DNA seems to end up in a waterway fairly routinely. They were detected at 152 sites. The Australian wood duck was detected at 158 sites, and one of the native water birds that we detected was the little pied cormorant, which was detected at 21% of sites, or 67 sites in total. Now, invasive species, unfortunately, we detected them throughout the Murray Darling Basin. Just about every single site had an invasive fish species present at it, as you can see by this figure, which gives you an indication of the proportion of invasives that were detected, with dark purple, I think it is, being 0 detected, all the way through to yellow being where the only things detected at that site were invasive fish species. There was a lot of different invasive fish species detected, 11 in total. Your usual culprits of gambusia, goldfish, carp, and unfortunately trout. Both rainbow and brown trout were detected and look like they're actually spreading across the basin, which is certainly a little bit concerning given their destructive nature, which can be made worse by intentional stocking. We also detected oriental weatherloach and roach in new waterways, which also may indicate that they're spreading within the basin. But in more positive news, tilapia, which is an invasive species that's been detected, particularly in Queensland, and it's a real concern that it might get into the basin. We didn't detect it at any of our sites that we surveyed, which is really good news at this stage. These are just pictures of your more common invasive species that were detected. Gambusia were at 138 sites. Goldfish were at 235 sites. So they were detected one of the highest invasive species across the basin. Carp were at 164 sites. And we detected trout. In this case, this is brown trout picture that you can see on the screen. Both brown trout and rainbow trout were detected at 62 sites, about 19%. We've essentially developed a data explorer, or a little web app that you can utilize to actually look at site results and determine all the different species that were detected at that site. This can be found just by scanning the QR code. It's also, I think, on the GORE's website that Odonata hosts, and it will be in the report and email that is sent out to you. So you can, if you haven't already accessed it, you can gain access to it. It looks like this. And what I'll do is actually just give you a quick overview of it. Just bring it up on my screen. You can all see that?
Yep.
Yep. So essentially, if you follow that link, you'll be brought to this page, which shows you the shading of the Murray Darling Basin and the various basins that make up the MDB in each of the different states. The dots in this figure represent sample sites, and they actually indicate the number of species when you first open this up that were detected at that site, with the little key on the left-hand side telling you the species richness. So I'll just show you that essentially you can zoom in just like you would on a Google Maps or any other type of online mapping tool. You can click on the dots, which tells you all the different species that were detected. The Actinopteri, which is fish, Aves, which is birds, and Mammalia, which is obviously mammals. It's got the scientific name, and then it's got in brackets the common name or the general common name. There might be other common names that you know the particular species by. There's a site code at the top, which is just the site number that we have that was sampled, that go with the coordinates that we were given. And then it's the results in the two columns representing 1 and 2 for the two eDNA samples. The actual numbers that you see tell you the sequence reads that were detected for that particular species in our data set. The more sequence reads generally tells you that there was more DNA from that species at that particular site. It is a little bit of a gross generalization, but that's certainly how you can interpret it. And so you can see in this particular sample, Bidianus bidianus, which is silver perch, a threatened species, was only detected in one of the two samples. And there was 158 sequence reads in sample 2. Whereas European carp was detected in both samples, and there was 36,000 in the first sample sequence reads and about 10,000 in the second sample, telling you that it's present at quite high amounts. You can click on any dot and it tells you exactly what was present. Move it around as you would any other map. You can also use the drop-down menus, filters on the right-hand side to zoom in on different either groups, being the different class, being fish, bird, frogs, birds, mammals, or reptiles, their threat status, so you can look at the different listed species, as well as you can search on individual species present. If you click a particular class first, being fish, you can then see in the drop-down menu just the fish species that were detected. And if you click on one of these, and I'll pick silver perch again, Bidianus bidianus, it will then bring up on the map all of the detections of silver perch from all of the samples. And as you can see, the various sites that this threatened species was detected at. Again, you can click on that, and because we have the filter on, it'll just show you what samples, so whether it was replicate 1 or replicate 2 or both of the samples that were taken at that site where silver perch was detected. One other feature of this this particular web app is that you can turn on ALA observations, so Atlas of Living Australia, so where the species has been detected previously. Just by clicking on that button on the right-hand side, it gives you the key for the year, and it plots all of the ALA observations for that particular species. And as you can see, the detections that we have largely overlap with what was known previous, although there is one detection up here that wasn't known previously. We also have VBA observations in the database. So this is the Victorian Biodiversity Atlas, so obviously exclusively for the Victorian waterways. And you can also turn on the negative site. So where a sample was taken and we did not detect silver perch. And that is largely the app. There is also 2 different views. You can also look at a terrain view if you want to go in and look at the site in particular a little bit more and understand exactly where that sample was taken from. If you do not see a sample that where you've taken a sample from a particular site, that could mean a couple of things. One could be that the sample had no actual DNA detected from the sample, or it failed our quality control process within the lab, and therefore the data is not represented, or it could simply be a mistake. And please, if you are worrying about a sample that you've taken from a site that is not represented on the map, then get in contact with ODNATA and we can track down exactly what's happened. And I think I will finish it there.